It was a project that we have run in Angola. It was a clinical trial with Hydroxyurea, and the idea was trying to see if we can identify some genetic markers that in some way can help the clinician to decide if the person will respond to the treatment, in this case Hydroxyurea. So we have found some genetic markers that in the future, in broader studies with huge populations, that it could be used to select people that will not respond to the treatment. It was a very interesting project because it was done in Angola, a low-income country in Africa, with several problems because sometimes the hospitals are not ready to conduct clinical trials, data collection, and it's sometimes difficult to follow up the kids because of difficulties of the mothers to have the transportation to the hospital. So we had to prepare all the setup in the hospital and in the research. We had a PhD student, an Angolan PhD student, she's a pediatrician that was responsible for the project. So we had found from a Portugal institution, the scientific foundation in Portugal that supports the study. So we had to prepare all the hospital for conduct a clinical trial and do all the study. The genetic analysis was performed by Angolan students in Lisbon, in Portugal, because we decided it was so expensive to start a new lab in Angola. So all the clinical part was performed in Angola with Angolans, and then with also Angolans it was performed the genetic parts in Lisbon. It was a challenge, but I think it was very, very nice, and we have other projects in Angola now working in that area also. We choose genes related to pharmacogenetic and pharmacodynamic of hydroxyurea that in other studies gave some information that they could potentially be associated. It was not an whole genome sequencing analysis, it was a focus for specific loci that we have chosen based in previous studies. The one most associated, it's a protein associated with cell adhesion. We are not right now sure about the function of the gene, but we found that since some of the problems related to sickle cell disease, it's cell adhesion, and it will go to some of problems in circulation. So probably there is association between the function of the gene and the phenotype of the disease.